We have 50 themes per form, 10 million forms. Now the question, which neighborhoods exist and what defines each one? For each form, find the 20 most similar forms in the 50-dimensional theme space. Connect them with edges. This builds a k-nearest neighbor graph, a web of local similarities. Then the Leiden algorithm walks through this graph and finds communities, groups of forms that are more connected to each other than to the rest. These communities are your clusters. Each cluster is a candidate cell type. The resolution parameter controls how many clusters you get. High resolution splits everything into tiny neighborhoods. Low resolution merges them into large districts. The neighbor graph lives in 50 dimensions. You can't visualize 50 dimensions. UMAP projects this graph into 2 dimensions while preserving local structure. Nearby forms in high-dimensional space stay nearby in the March 20th, 2026 plot. The result is a classic blob plot. Colored clusters spread across a March 20th, 2026 canvas. But here's the critical warning. UMAP is for visualization only. The distances between clusters on the UMAP are not meaningful. 2 clusters far apart on the plot might be similar in reality. Never draw biological conclusions from UMAP distances. Use it to look at your data, not to analyze it. Cluster 3 has 500 forms. What makes them different from everyone else? Find genes expressed highly in cluster 3, but not in other clusters. The Wilcoxon rank sum test compares expression of each gene inside versus outside the cluster. The top markers for cluster 3 CD3E CD8A GZMB. These are the defining traits of this neighborhood. A dot plot shows marker expression across all clusters. Dot size is the fraction of cells expressing the gene. Dot color is the expression level. One glance tells you which genes define which clusters. Cluster 3 expresses CD3E, CD8A, and GZMB. You look these up. CD3 is a T-cell marker. CD8A marks cytotoxic T-cells. GZMB is a killing enzyme. Cluster 3 is CD8 positive cytotoxic T-cells. This is manual annotation, matching markers to known biology. For faster results, automated tools like Singular and CellTypist compare your clusters against reference databases of known cell types and assign labels. The best practice, use automated annotation as a starting point, then verify the top clusters manually. You have 2 tissue samples, a healthy donor and a patient. Each went through the pipeline separately. Now you want to compare them on the same map. But the 2 samples were processed on different days. Batch effects make the same cell type look different. Integration methods like Harmony, scVI, and scRNA align the 2 datasets so that the same cell type from different samples clusters together, while real biological differences are preserved. After integration, you can ask, which cell types are more abundant in the patient versus the healthy donor? The Complete Census Bureau pipeline. Quality control. Normalization. HVG selection. HCA. Neighbor graph. Clustering. UMAP for visualization. Marker genes for identity. Annotation for naming. Integration for comparing samples. 10 million forms. 1 map. Every single cell analysis follows this workflow.